veterinarians who are volunteers and members. I want to thank all of you for helping support the animals, for your time, for helping take care of the animals, and also for your donations as members and supporting the center. It's, as most of you have probably heard, it's been a crazy time this year at the center. I wanted to give you a little bit of update. As you saw outside on the board, we currently have 170 animals on site, but yesterday we released 12 sea lions up in Cindy Rock, so we're trying to get them out as fast as they're coming in, but we've sustained over 150 animals on site since the beginning of April into March, which is just unprecedented for us. So far this year, we've rescued and we've brought in 484 animals up to 2 days ago, which is incredible. All of last year, I think we did 430 animals. We've already surpassed the number we did last year. And in our normal average year, we see about 600 animals here. So we're closely going to hit our average, and depending on what's going on out in the environment there, pending El Nino, this could be a really busy year for us. But luckily we have lots of supporters and members like you that will help us take care of all these animals. I would like to thank Laura, our events manager, in the back, for setting this all up. Also our communications manager, director, who who helps manage all the media that comes through here. And I don't know if you've seen this in the news, but the last couple of weeks has been a big story with the media. So thanks to Laura for all the things that she does here also. And we also have one of the, we are a rehabilitation center, but one of our missions is also research to learn about these animals. And we have a special guest here tonight who was a volunteer back in 1990, 1998, and was actually a supervisor on Wednesday night crew, and that's Dan Fletcher back here. Say hi, Dan.
Smelly guy in
Dan was a volunteer here, and it changed, and he's an engineer, and he's volunteering here with animals changed his whole career path, and he went back to vet school and became veterinarian, and then he became a specialist in critical care medicine. And he was just visiting last year and talking with Francis and myself about the Otis triangles cases and how we just have no good treatment for them and how they bleed out and from the worm and infection. And Dan is studying these new medications in dogs and other companion animals to treat bleeding disorders. So we came up, Dan wrote a grant, got some funding. He's brought one of the veterinary residents here, and they're here this week. We're testing some drug levels on some of our elephant seals and see if this might be a new way to help care for our Otis triangles cases. So it's great to have Dan here. And, we're striving all the time to find better care, for our patients. So, but tonight we're going to talk about a more global picture, tracking marine mammal health, and we'll hear all about what this really means. But I think you'll find this a really interesting topic, and I would like to introduce Dr. Claire Simeone. She's going to be telling us about this new project that's been piloted by the Marine Mammal Center and Francis Golan and the workshop that we had here last month. Claire is the Conservation Medicine Veterinarian here at the Marine Mammal Center. She started last year here and it's a joint position with the National Marine Fisheries Service and the Marine Mammal Center. The position is actually funded by NOAA, but the position lives here at the Mammal Center and works closely with Francis and Terry Rolls at NOAA to train veterinarians to understand policy and the big picture about marine mammal health in general. And Claire came from us from her internship was most recently down at the Navy Marine Mammal Program in San Diego. And before that, I did that internship in San Diego and went to Virginia, Maryland. Is that right? Yeah, for vet school. And so she's been traveling all over the country with Francis and going to meetings and learning about marine mammal issues. And will tell us about this new pilot program that we started earlier this year. Great.
you're here to listen to this, because I think this is really a passion of mine. I'm very excited about the program and really how it takes a holistic view of what's happening to marine mammals, what's happening to our oceans. And so I'd love, as we go through, if you have any questions, we can certainly talk about them then, or we'll have some time at the end. But let me know if you can't hear me or if I'm going too fast with anything. Does it work? Is it on? Is it on? It is on. Oh, it works. That's okay. I'll use this little button here as we go. Okay. So I want to take a look at the holistic picture of marine mammal health and ocean health. And one of the things that we typically get questions about is ocean health deteriorating? And from fisheries to marine mammals to the actual water quality, it's a big question. I don't know if you all are aware of the Ocean Health Index, but it's a program that was started to evaluate country by country the health of the ocean. But one of the things that's really missing is that it doesn't include any health data, surprisingly enough. So it doesn't include any data about marine mammals, fish, anything like that. And we know that marine mammals are an extremely integral part to evaluating the health of the ocean. And so we know that in the past several years that there have been an increase in the individual numbers of animals that strand, and you can see that right at the center. There have been an increased number of publications on marine mammal health. And then there's also been an increase in the number of unusual mortality events. For instance, right now there are 8 unusual mortality events around the country that are currently open that we're evaluating of groups of animals that have stranded and died. And if we take a look at our admissions to the Marine Mammal Center, this is the number of animals that were admitted to the center since the start in 1975, and it's through 2009. But you can tell that there's an increasing trend of the number of patients. And as Sean mentioned, we know that this year is a record year for us. We have a variety of health issues that are affecting these animals that come in.
The lower numbers, could that just be because there wasn't an awareness of what we were doing in the center, and maybe there were just as many animals becoming ill, and we just didn't know about it?
That's a great point. So some of it absolutely could be a lack of response. And if you look at other centers around the U.S., this is a world-class, very respected center. Some of the other locations, there are either no marine mammal surveillance, or the response is just a few volunteers, kind of like it was in the beginning. And so that certainly could play a role. But at least if we look back in the past 10 years, we have seen increases where our standard of care and response has been pretty level. If that makes sense.
Yeah, thank you.
Mm-hmm. And so one of the things that I'm working on right now is looking at a review of all of the marine mammal literature, health literature that's been published since the passage of the Marine Mammal Protection Act in 1972. And that was really an integral act that helped revolutionize the way that the awareness around these animals and protection for marine mammals in general. And so I have split off by region the number of published sources by a variety of categories. And the size of each pie chart corresponds to the number of publications in that region. And one of the things that we can really be proud of is that if we look at California has a very large number of publications in compared with the rest of the United States. And the Marine Mammal Center contributes to a very large number of those publications. And so the things that we're learning about the animals from the center contribute to this whole body of literature that tells us, not only about infectious disease, but also about contaminants and cancer and biotoxins, things that we'll talk about in just a few minutes.
What are non-oil contaminants? They're pretty big involved.
So I split this out. The oil is, what the oil is. Non-oil contaminants is really a catch-all for PCBs, organochlorines, anything else that typically tends to be lipophilic, so it stays in the blubber of the marine mammals. And so because they are they tend to be apex predators, the contaminants that come into the fish that they then eat go into the marine mammals, and they stay there for a very long time. So we're able to see, we're able to see contaminants that were banned 20 years ago, and they're still in the blubber of these animals.
Why did you separate out urogenital carcinoma from other neoplasias?
So urogenital carcinoma with sea lions is something that we see very commonly here at the center. And I'll talk about that in just a couple of minutes. Other types of cancers are actually rather rare in comparison with other species. And so this was really highlighting that other than the urogenital carcinoma, which we know has many different factors that contribute to it, cancers tend to be a much smaller proportion of cases in study literature. And so we all know, we're very familiar that human interaction is a leading cause of damage to these animals. And we can see that an animal here on the bottom left that was covered with oil. Animals that have been shot with arrows or gunshots. These are all pellets that when this animal was shot in the head, and so many entanglements. And so human interaction, I have split up the trauma cases into trauma that we are responsible for, as well as then natural types of trauma, like shark bites or things like that. Because this really is very telling that we are responsible for a large number of these cases. And so I don't know if you all are familiar with domoic acid. It's a toxin that's produced by algae called Pseudonychia, and it does affect humans. It causes amnesic shellfish poisoning in humans, and we can see two types of effects in marine mammals, and marine mammals are certainly affected by it. When animals eat a large proportion of toxin in the fish that they eat over a short period of time, they can have an acute toxicosis. And that's characterized by seizures, coma, necrosis of the brain, specifically of the hippocampus. We know that is the part of the brain that it attacks. However, this is a naturally occurring toxin, and so we know that there are low levels in fish. We know that there are low levels, and with chronic exposure over a prolonged period of time, you can still get changes to the brain. And they actually cause anatomic changes that are long-lasting and sometimes can cause some very severe effects. We know that it manifests itself as unusual behavior in a lot of these animals. So, whether it's overt aggression, some of these animals are very aggressive. Some of them are very lethargic and basically immovable. Or sometimes they'll go in places where they shouldn't go, on top of police cars, or in people's cars or in their backyards, and so it's often a kind of a funny story. But we know that the reason that they're doing this is because they have permanent brain damage from this toxin. And one of the things that we are lucky enough to be able to do in certain cases is to actually look at the brain with MRI. They can go to a machine in a human hospital, and then we can look and we can see, this is a picture of a normal brain, and then we can actually see the part of the brain that's called the hippocampus that is abnormal. And I think there was a better picture of it, one back. Oh, wait, maybe not. Where that part of the brain actually looks shrunken and atrophied. And we know that the toxin over prolonged exposure causes that atrophy to the brain.
What is horn damage?
That's just the portion of the brain, the temporal horn, that becomes enlarged when the hippocampus itself becomes reduced in size. So those are both effects.
Does that affect the amygdala too?
Yeah. It does not specifically affect the amygdala. And so, as we had talked about before, with cancer, about 17%, and actually it's now increasing, I'd say it's closer to 20 or 25%, of the adult California sea lions that we see and we're able to do postmortem exams on will have this urogenital carcinoma. And it originates in the cervix in females and the penis in males. It's a cancer that affects the reproductive tract. And death is typically due to metastasis or spread of the cancer. So sometimes the tumors will go to the lymph nodes and they'll get so large that the ureter is blocked. And so the urine blocks up and it causes kidney failure. Sometimes the cancer itself goes to the lungs or the liver. And this is a picture of that this really, all of this here, this white that you're seeing is all cancer. And so we also see a variety of infectious diseases. One of that is kind of prototypical here is leptospirosis. And it's a bacterial infection that's caused by the bacteria Leptospira interrogans. And there are many different serovars or different strains of the bacteria. But the one that most commonly infects sea lions is the serovar Pomona. But Leptospira is distributed worldwide, and it affects humans. And they see about 100 cases per year in humans in the United States. And it's something that's actually emerging in California, meaning that they're seeing an increased number of cases every year in humans. And so what happens is the bacteria live in the kidneys, and then they're excreted in the urine. But they can hide really well. So we can treat it with antibiotics, but oftentimes it doesn't penetrate to the kidneys well enough to kill all the bacteria. And so animals can shed the bacteria that are infected for years. And we do see that with certain strains of bacteria, if they are used to infecting a certain host, then you'll have kind of low levels of disease and it'll sort of smolder in the population. But then if you have a serovar that is abnormal and isn't used to being in that species, then you'll see epidemic disease, where you'll see these regular spikes of high mortalities and very, very sick animals. And we see those epidemics in California sea lions with some regularity. This is a graph that shows in the background in the black, you can see the total number of deaths. This is for animals at the Marine Mammal Center. And then in red, you're seeing the spikes of the number of deaths that were attributed to leptospirosis. And so we see that about every 3 to 4 years, there's a spike. And then there's some cases in the background and a spike. And that's the epidemic disease that we're seeing for these non-host adapted species.
These sea lions gather in large groups at Pier 39 and Bodega Rock and lots of places. Why doesn't every one of them have leptospirosis?
That is a great question. So we have there is a PhD student and a postdoctoral student down at UCLA that are studying leptospirosis, and they're studying it specifically in sea lions out on the rookeries. And we know that leptospirosis in sea lions acts. It doesn't really act like a host adapted species, and it doesn't really act like a non-host adapted species. We see these peaks every few years, but then we also see these smoldering cases on and off. And so part of it is that animals that get sick but then survive have an immunity that's usually lifelong. And so then it's the younger animals that aren't exposed when they're actually on the rookeries, where it's like Pier 39, where you have just a mountain of animals on top of each other. And then it makes it very easy for that bacteria to spread very quickly. So every once in a while, when the concentration of the bacteria and the number of animals with no immunity gets high enough, then you see that big peak. Does that make sense?
It's a freshwater organism. It comes down rivers.
It is. And the other thing is that we don't know where it's hiding itself on the Channel Islands. And so these are all questions, ongoing research that's happening. Yeah. Interesting. So in addition to these known cases, we also have unusual mortality events, which we shorten to UMEs. And these are mass strandings or die-offs. Typically the animals do die, but sometimes they're just sick. There are a lot of sick animals for a reason that we don't know. And there's a working group of experts that comes together, a panel, and they evaluate these requests to have an unusual mortality event declared. And they look at the data and they decide, is this unusual? Does it garner a lot of attention? Should we put specific funds into studying that? The question that's very difficult, though, is that for a lot of these species or populations, we don't have baseline data. And so we don't know what usual is. And it's really difficult to tell what unusual is if we have no idea what usual is. And so that's one of the big problems with UMEs. And there are a couple, I said before that we have 8 that are active right now. A couple that are going on that you may have heard about in the news. There's one on the mid-Atlantic coast, and it affects bottlenose dolphins. So since last June, more than 1,200 animals have stranded from New York all the way down to Florida. And luckily, we have tests for morbillivirus, and we were able to diagnose very quickly that it was a morbillivirus outbreak that was happening. And morbillivirus is similar to measles virus in humans and canine distemper virus in dogs. And unlike the leptospirosis, where a lot of animals have this long-lasting immunity, it doesn't act similarly to that, in that we see that the immunity drops very quickly. And then you have these entire populations of animals that have no antibodies to morbillivirus. And at that point, we're not sure exactly where it comes in, but when it comes in, it explodes through the whole population. And we see very severe pneumonia. We'll see that it affects the brain. They get meningitis, and they'll have neurologic signs. And unfortunately, when they don't have antibodies, they tend to die very quickly. And so that's one part of my job, is that when I work with NOAA, then I'm sent out to respond to unusual mortality events like this. And then another thing that's closer to home that I'm sure you all are very aware of was this UME in California that started last year. And in Southern California especially, but up and down the coast, California sea lions that were very malnourished were stranding on beaches. And we looked, the working group had an entire plan to rule out different infectious diseases, and we found that the animals did have a variety of infections. They would get pneumonia, they would get enteritis, an infection of the GI tract, but there wasn't really any kind of overwhelming infection that was characteristic among all of them. And the great thing about the UME working group is that it brings together experts that aren't not just veterinarians, but they're epidemiologists, and they have expertise in oceanography, and they're fisheries experts. And we were able to have a more holistic view of what was going on. And because we collaborated with fisheries experts, we were able to discover that the sardines that they typically are feeding on had moved offshore. Not due to overfishing by humans or anything, but likely due to changes in currents and that they were moving offshore. And so the pups' moms had to go further to forage, and so they were exhausting themselves as they went. And so a lot of the times they were just abandoning their pups. And then the pups, that when they were on the Channel Islands, they would go out to forage, and there was no fish there. And so we were seeing these animals that were stranding along the beaches, and that we're still seeing now. But this is one of the few UMEs that we actually were able to determine a cause from that, thanks in part to that large collaboration and having all that data. And so the Marine Mammal Center really plays an integral part in all of this. In all of those different cases, we have veterinary staff and stranding staff, all of the volunteers that rescue the animals and care for them. And I mean, if you think about the sheer volume of animals that come through the center, we have an incredibly large amount of data from each of those animals. And for instance, so I'll show you this video of a harbor seal named Agaptimos, and he had cataracts in both of his eyes. And we had some researchers at UC Davis that built an enrichment box for him. And they could put the fish in the box, and then he would have to lift up the little flap and take out the fish. And we found that even with his cataracts, he was able to find the fish just as well as all of his other little friends. They're very cute. And it's really great for them while they're in rehab, because they have to use their lungs. They have to stay down on the bottom longer to find the food. They have to use their muscles. And so it's really great enrichment for them. Except when his friend steals it. But it's also really great for us to be able to evaluate the animals. So if you think about Agaptimos, and he's just one animal, but already we have an incredible amount of data on him. We've learned so much about his physical condition, about how he's going to survive, and about his health in general. So then if you think about that, if you think about all the animals that are coming in, and then you multiply it by that by the entire country. So you may be familiar with the NOAA, NIMS, and then the HSRP, of course. And that stands for the National Oceanographic and Atmospheric Division, and then there's a branch called the National Marine Fisheries Service, and then within that is the Marine Mammal Health and Stranding Response Program. And that is the government entity that oversees our work, and they provide us with the permits that allow us to do our work here. And so we are member organizations under the Health and Stranding Program. And so if you look at California, we have a very large coastline that we respond to, but we also have 7 other facilities that are also doing the same thing that we are. And they're seeing animals that are coming in, and they're collecting that data as well, those data. And then if you expand it out, we're just the West Coast region, and we have the Southeast, the Caribbean, the Northeast, the Pacific Islands, and Alaska. And there are multiple stranding centers in each of these regions. And yet, we have all of these data, and yet we don't have a standardized system to report it. And so we have about 30 years of data that we don't have access to a lot of the times. And there is such a need for responding to the UMEs, to having that baseline and knowing what's normal. We really needed access to this. And then on top of that, we're just talking about the stranding network, but what about the healthy ones? We have biologists that are going out and they're studying the populations, and they're looking at the number of animals that are in certain locations, how that's changing over time, looking at contaminant levels. For instance, this is a group that I worked with up in Alaska. They were looking at stellar sea lions. And in the western Aleutians, the stellar sea lion population has been declining over the past several years. And they don't know why. And they found that a certain specific group on one of the islands in the western Aleutians has an extremely high level of mercury, and they do not know where it's coming from. And that's the first time that they've really been investigating it. But they're taking all of these samples, and they're counting all of these animals, and that really should be included as well. And then you think about ecotourism, that members of the public, if they're going out on whale watching boats, they're counting animals. That could certainly be part of the data set. Animals in aquaria, collection animals, they provide a large amount of data about their health and about new techniques and new advances in marine mammal medicine. And even law enforcement, they will get reports of nuisance animals, and a lot of times they come to us, but some of the times in different states and everything, then they respond to those cases. So there's an even larger amount of data that can be used just with the healthy population as well. So we wanted to put it into a map. And we had a lot of discussions about, what the platform should be, how it should be introduced. But we know that mapping isn't new. It's kind of the hot thing right now. And there are a variety of programs that are out there already. One of these is healthmap.org, and this is with Harvard University. And it actually uses the crowdsourced data that I talked about before. So it scans Google. It's a little scary, actually. It scans Google, and if you've said, I think I have the flu, then it counts that. And so then it puts together these hotspots of where potential outbreaks are occurring, and it does it in real time. So it's not just waiting for the CDC to be reporting specific cases. It's tracking it real time. It's a pretty impressive program. And then actually, within healthmap.org, there's a program that's called Predict. And this is a USAID project. It's out of University of California at Davis, whom we collaborate with quite a bit. And it is looking at disease outbreaks with pathogens that are most likely to be possible transfers to humans. So it's looking at emerging viral diseases with species that we know have the potential to mutate. And so that includes looking at viruses in primates and in birds and in rodents. And so then it has people in the field globally that will go out and sample populations, and then it will study all the viruses that are intact there. And they've actually discovered a number of new viruses that people didn't even realize were there. And they've discovered yellow fever in a species of monkey and so they were able to actually show that an outbreak of yellow fever was likely in that area just by doing these routine swabs. So PREDICT is another fantastic program. One that's also on the wildlife front, but looking at terrestrial wildlife, is called the WHISPERS program, and that is through the National Wildlife Health Center in Madison, Wisconsin. And they look at outbreaks. So when a group of more than 5 animals die, then they'll report it here, and they break it down by the different types of disease. So here we're actually looking at white nose syndrome in bats, which is a fungal disease, and we're still learning about it. We still don't really know what causes the bats to have the fungal disease. And so it plots it on a map so that wildlife managers and people that really need to know where these pathogens are spreading are able to look at these type of data. So, our vision, looking at all of these, was to have a centralized data reporting system and really eventually expand it to a national database and even an international database one day. And to really capture all aspects of marine mammal health that we went over, all of the different diseases, all of the healthy animals, everything. And that is a huge undertaking, obviously, so we had to start small. We had to start somewhere, so we decided that we would do a pilot project. And so the Marine Mammal Center, along with other collaborators in California, other rehab centers, all pooled our data together. And this happened this winter. And so we had Southwest Fisheries Science Center, the Marine Mammal Center, the SeaWorld Rehabilitation Program, the Marine Mammal Care Center at Fort MacArthur, and the Santa Barbara Museum of Natural History. And we looked retrospectively, so we looked at dead animals that had come into these facilities between 2005 and 2010. And we know that because they're all part of the Marine Mammal Health and Stranding Response Program, that they collect a basic level of data that's called the Level A data. And that includes the age, the age class of the animal, the sex, the length, and then the location where it's stranded. So we have latitude and longitude, so we can map it really easily. And then we also included a health category, and we assigned the cause of death to usually one, but sometimes a variety of categories. And those categories were broken down kind of into the groups that we talked about earlier. So biotoxin includes domoic acid, and then in other parts of the country you have different types of toxins that affect marine mammals. We broke down infectious disease into 2 categories. So known pathogens like leptospirosis went in one category, but sometimes animals will come in and they'll have pneumonia, but we don't necessarily culture it, and we don't figure out exactly what the bacteria is. And we say, okay, yes, we still want to capture that, so they would go into the non-specific infectious disease category. If animals were just malnourished and we really didn't find another cause of death, they would go into the malnutrition category. Neoplasia is any type of cancer, and in our study it was almost exclusively urogenital carcinoma because that's what we see along the California coast. And then, as I said before, the trauma was broken down into human interactions, like the fisheries interactions or other types of trauma. And then sometimes animals, sometimes we will get dolphin or whale carcasses that are very decomposed, and we don't know what the cause of death is, but they still stranded. So they would go into an unknown category, or other if they had something strange like kidney stones that didn't really fit in anywhere else. Oh, and so this is another one of those types of pie chart maps here. And again, the size of the pie chart corresponds to the number of cases in each one of those counties. And so, as you can see, some of the counties only see one aberrant sea lion that runs the wrong way, and then some of them, like Monterey Bay, actually have hundreds of cases just over the 5 years. But you can see that there are nearly all of these categories in every county. There are some differences in the way that they're spread out, but we see a variety of these diseases at every stranding center. Yeah?
Which county is the one that has the biotoxin?
Yeah. You're going to test me on my county. All right, let's see. This one.
Yeah, that one.
That is Is that San Luis Obispo?
Is it San Luis Obispo?
I think.
Oh,
That's San Luis Obispo.
All right.
That's right.
I don't know if that's the name.
That's San Luis Obispo. And we do see in San Luis Obispo and also in Monterey that a large number, we'll get these concentrations of toxic blooms, and then we'll often get a large number of animals at once that are affected by those blooms. And I think that really the reason that we don't see a larger number of biotoxins in Monterey is because we see such a higher number of animals in a variety of cases. Going in, because the stranding centers don't typically capture a lot of contaminant data, usually those are biologists that are going out and sampling, or if we do a study on mercury, we'll send them off to a different researcher. And so for this pilot study, we didn't include any contaminant data, but the hope is that that will be included in the future.
Was this just data that came through rehab centers?
Yeah.
Because there's other data sets where you have like beach watch programs or Cal Academy of Sciences that have years of data of beach data anyway.
Absolutely, and that is really going to be very important when we talk about the baselines.
Yeah.
Is that we've been trying to figure out what is the best way to qualify a lot of these data. Is to say, did somebody just happen on a carcass, or is somebody combing the beach every day and not seeing them?
Yeah.
Because the absence is
just
just
so
as
in those
important.
areas from San Mateo up to Sonoma, you have 25 years of data that each beach is looked at at least once a month.
Absolutely,
So
absolutely.
yeah.
And so so they will.
And that would make those
definitely
pie charts a
draw
lot
in.
bigger then, because
Yeah.
that's not true down in Morro Bay area. for example.
Absolutely, yeah, that's a great point. And so, that map was pretty cool, but we wanted to do better. So we wanted to look at different platforms where the Marine Mammal Health Map could live. And we went through a variety of options, but one that we settled on was the Integrated Ocean Observing System, or IOOS. And there was actually an IOOS bill that was passed. And so it created these different regional offices that collect all of the oceanographic data, a large number of the oceanographic data throughout the country. And so they will track things like sea surface temperature, salinity, chlorophyll levels, and they're divided by regions. So it worked out very well for us because they have the Southern California Coastal Observing System, or SCOOS, and then they have the Central and Northern California Observing System, or CENCOOS, and they're split up sort of exactly where our data set would lie. But it's easily transferable between regions, and you can layer. We could layer our data set on top of this existing data. And one of the things that's really cool about IOOS is that they have these models that they put in. So here is this is showing you the probability that domoic acid toxin is going to be present. So one of the things that's really interesting about domoic acid is that we don't know exactly why the algae produces the toxin. Sometimes you'll get a huge bloom and there will be no toxin. And then other times you'll get a small bloom and it'll be very, very toxin concentrated. And so IOOS is one of the first people they're using their existing temperature and salinity and chlorophyll data, and they're modeling it to figure out what the probability is that that algae will produce that toxin. And one thing that's really striking is that this is from March 26th, and you can see that right around Monterey Bay there's a hot spot there. And that correlated very well to the large number of seizing sea lions that came into the facility then and in the weeks after. So it could potentially be an extremely useful tool, especially because sea lions tend to get sick before a lot of the mussel species that we actually test for even show up positive for the toxin. And so the human health monitoring is constantly checking mussels to say, we need to have a warning. And they've actually shut down, they've had a public health warning now that you shouldn't eat mussels or shellfish or anything in this area. But we were seeing that the sea lions were getting sick before they were even detecting levels. So oftentimes we'll tell the surveillance people, we're getting a lot of sea lions, and you should watch out because the toxins are going to be rising. So it's a really beneficial relationship. And so, this is what everything looked like for the pilot study. And I'm going to play a little video going through it, but just to sort of orient you here, we put all of the data points into hexagons. And so if you look, there are varying shades of red, and the ones that are very brilliantly red colored have a large number of individual data points within that hexagon. And then the ones that are more see-through may only have a couple of points in the hexagon. And then, let's see, I'm going to try to do this here. So as we go through, so you can scroll over each of the individual hexagons, and it will tell you the number of cases broken down by category. And so you can see that in that hotspot on Monterey Bay, that you have 219 cases of malnutrition, you have 195 cases of non-specific infectious disease. And this is for the whole time, that whole time of the data set. And then it also breaks it out by the scientific name, so you know what species were affected then as well. And then if you actually click, if you click on one of the hexagons, then it brings up a histogram or a bar chart here. And that actually, as you scroll over it, it'll tell you through time the number of cases at that point. And so you can see that in 2009, in the summer of 2009, we had a large number of sea lion strand. That there were 54 sea lions just in that time period alone. And so it really gives you a way that you can evaluate the data both spatially on the map as well as temporally, which makes it really, really valuable data. And so you can scroll through, you can close that out. Then along the bottom here, you can see that everything is broken down by time. And you can actually close the data set by a certain amount of time. So you're looking at just a month, and then you can move it, and the data appear as you move. So you can
Is there any correlation to like El Nino years built into this plan?
That is, you're like totally right on track. That is exactly what we want to look at. Yes, we'll talk about that in just a second. So yes, you can look and see how the strandings occur as we go through time. And then you can filter by health category. So if you just wanted to look at the biotoxin cases, you could see the species that were affected at that point. And you can also break it down by scientific name. So you can look at just the harbor porpoises or just the California sea lions. So here, we're just choosing the harbor porpoises, and then you can see, and interestingly, that hotspot in San Francisco, we knew that several years ago there were a lot of harbor porpoises that were being killed by dolphins, and they came in, and so those are the trauma cases, if you break that down. And then you can also use a polygon tool, so you can freeform draw around the hexagons that you want, and then it'll pop up the little histogram again, and you can see, okay, so in my area of interest, so just San Francisco Bay or just Monterey Bay, this is the number of cases that I'm seeing. So that's a really useful tool. You can zoom in exactly what you want. As you zoom out, the number of hexagons gets smaller, but the number of cases in each individual hexagon gets larger. And so you could scale out to the entire country once we have that data set. And then you can also scale in, so you can look at just Morro Bay, and you can look at the number, then you look at the number of actual individual cases that are stranding at that latitude and longitude. And so if we had a hotspot with a large domoic acid bloom, you could see exactly where those animals were stranding.
That's fantastic.
Isn't that cool? It's like very much cooler than my graph that I showed before. So this is, so it was actually through, so CENCUS, the Central and Northern California Ocean Observing System, has a group that works on their data set called Axiom Consulting, and they're based out of Anchorage. And we gave them our data set. Tanaya and I Tanaya mainly worked on this for like months, and he put everything in 3 days, and he was like, I'm done, that was easy. And we're like, okay, thank you. And one of the reasons that it's so easy for them is that they have all of this pre-existing data. So we're pulling up sea surface temperature here. And you're looking at each one of these little pixels is a sensor in the ocean. And so you can click on one of the pixels, and it brings up the variation in time and the fluctuations of the sea surface temperature. So we'll make that bigger. And you can see that you can look for, okay, is this the normal oscillation of the temperature, or was there a sudden spike in temperature? If you're seeing an El Nino event, And so it's really a fantastic way. I mean, they have millions upon millions of data points, and we gave them like 4,000. So they were able to do it very quickly, but then we have access to all of that existing data. So it really makes our analysis so much stronger. It's phenomenal. So we were very happy about that. So it brings us back to this big question of whether or not ocean health is deteriorating. And marine mammal health is going to give us a big key as to how that interplays with all of the other oceanographic factors. And we've talked about so many different aspects of things that could be potentially part of the data set, and that could give us that information. And so you had asked about El Nino, and that is something that we're looking at next. So, first we're of course looking for funding sources. We would love it for it to be funded multi-years so that it can become this national program. And we're working with NOAA and with the stranding program to help to adopt this to be a national program. And we actually, Francis and I were in Washington D.C. last week, and we met with several Congress people who are very interested in incorporating it into different bills like the reauthorization of the IUS bill, things like that. So we're hopeful that they're going to take an interest in this and see how important it is to really be a key player in this. But one of our continued studies is that we're going to look at El Nino. And so we're looking at our events to see how the sea lion pup strandings, if they had anything to do with El Nino. And I'm not sure if you all are familiar with exactly how El Nino works. It's really complicated, and I'm very much simplifying it. But basically, if you look at this, so we're looking at here at North America, South America, Asia, Australia, and this is the Pacific. And this band right along the equator is what is involved with El Nino or La Nina events. And an El Nino conditions basically just mean that the sea surface temperature at that band along the Pacific gets warmer. And when it gets cooler, that's a La Nina event. And they have to, in order to officially declare an El Nino event, you have to have repeated increases in temperature, and they have to document a lot of other things. But we know that the shift in temperature basically means that this nutrient-rich upwelling from the Pacific that provides a lot of the phytoplankton is not there because the temperature is higher. And so this means that there's less for the fish to eat, and so the fish will go to different places or they just won't survive. And then that means that there's less fish for the marine mammals to eat. And so we know that years that are affected by El Nino, that we see an increase in strandings, and we're they are on the brink of saying, yes, this is a very significant El Nino year, and so we're expecting that in the next 6 months to a year that we're going to see these increased strandings. And so one of the things that we're hopeful is that we can look at our data set and we can compare it with the oceanographic data, and we can say, okay, we knew that these factors were happening before we declared the unusual mortality event. And so can we then move forward to predict repeat events and so that we can, one, funnel money and resources towards responding to these animals, but also look at what we can do to, possibly mitigate things and respond in a more efficient way.
That is fantastic.
Thank you.
This is great.
Well, why are you just looking at sea lions? Because I think El Nino's affected us with elephant seals even more so than sea lions.
Yes.
Yeah.
So one of because we have the unusual mortality event working group, and we have that data set with multiple stranding facilities, we're able to look at that a little bit more easily, and especially because the UME has an increased significance for the scientific group. But you're absolutely right that it's not just sea lions that are affected by it, for sure. Mm-hmm. And it's not really just important for marine mammal health, but there's a huge link for human health. We talked a little bit about it before, that domoic acid, that the sea lions are early indicators for domoic acid intoxication, and that we work with the people that are doing the muscle surveillance to help with really determining the levels of domoic acid. If you take a look at the Arctic, the food safety becomes a more direct issue, because it's not just affecting the fish that we're eating, but subsistence hunters are actually eating the marine mammals. And so when we have, there's an unusual mortality event in Alaska going on right now where animals are losing their fur, some animals are very sick, and we don't know what's causing it. The questions that people are asking is this safe to eat? And we don't know the answer to that a lot of the time. And so it's extremely important for public health. Again, in Alaska, if we look at sea otters, with the Exxon Valdez spill, we knew, there were immediate effects to otters and birds and other wildlife. But we know now that the populations of sea otters are just now starting to come back to normal 20 years after the Exxon Valdez spill. And so these contaminants have an extremely long, not just effects in wildlife, but extremely long effects on populations and everything. And if it's affecting them that way, it absolutely has the ability to affect us. And then climate change, is definitely the big elephant in the room. And Obama released a large report on climate change just this past week. And we know that that habitat loss is something that's extremely relevant for marine mammals. For instance, monk seals, we're opening our new hospital in Hawaii just this summer to take care of monk seals that are critically endangered, and we know that about 40% of the breeding grounds that monk seals use have been already destroyed because of sea level rise. And so it's something that's an immediate threat to these animals, but we're not far behind to be affected by that as well. And so it seems like a lot of gloom and doom sometimes with a lot of different pictures and everything. So how can we help? So first, keep doing what you're doing, keep being awesome. It's because of people like you that are members that are either donating their time or money, all of your hard work. We would not be a center without all of the volunteers and everybody who are contributing to that help. And so we absolutely appreciate it in the ways that we care for our animals. And then, maybe explore new support, new ways to be supportive. If you haven't considered being a volunteer, either with animal care or with education, or if you haven't considered, assisting with, there are so many other great causes that even just picking up trash on the beach here, that makes a significant difference. If you're politically minded, you can contact your legislator because they really have the power to funnel a lot of funds towards big projects like these. And so that's something that is always important. We're very lucky that our legislators in California are totally on board already. They say, yes, we absolutely need this for our ocean health. And then, bring us your ideas, because if you work for Google and you know nothing about marine mammals, but you know how to make a really cool app, or if you're a chef at home and you think of a new way that we can make fish milkshakes or something that we haven't thought of before, then that's that's extremely valuable, because we need, just like in the unusual mortality event working group, we need these experts from all different fields to tell us, to put the light bulb on and say, have you thought of this before? So we always love to hear from everybody about different ideas and novel ways of addressing these issues. Because it is just a really holistic problem, and something that we need to work together on to solve. So I always love hearing from people. You can feel free to email me if you have any questions about anything, or I'm happy to talk to you after here, and we can take questions now. Thanks for listening.
You know what? And once you finish your model, they can use it all around the globe.
Absolutely. We did the pilot study, and we're hoping that same thing will happen in the U.S. and then spread to different areas. We have a veterinarian from Chile, Dr. Mauricio Ulloa. Oh, hello. There. And he's training with us for 3 months. He's training with us for 3 months because he's in charge of the stranding network down in Chile. And so, it's always something that can be expanded and utilized for all kinds of response globally. Absolutely.
This is fantastic. Thank you.
Who taught you PowerPoint? I want to know how to do that.
Oh, it's Prezi. It's really cool.
That's a lovely presentation technically as well. Thank you.
Thank you very much. Rachel, Jack, yeah.
haven't had a chance to talk about this, but I'm wondering if there are any thoughts around using the public to help, or making this data set, when it's available, open to the public for citizen scientists to, look around, tinker, and have, ask their own questions and pose questions using this data set to answer some of those questions about, or look for trends and that sort of thing. So awesome to have, marine mammalogists and biologists working on this, but what about scientists from other disciplines or even the public? Absolutely. To look at the data and answer some questions.
Absolutely. One of the things that's wonderful about IUSE is that part of the act means that all of the data that's on the website has to be made public to everybody. And so that's really part of it, is that we want to make this visible not just to everybody else that we're collaborating with, but to the public, so that they can say, like, you see a ton of sea lions or harbor seals or elephant seals. And so it's going to be just as valuable for people to see, and for us to see something that's actually really visually appealing, and where people are like, that is really cool. Like that makes a difference in showing the data, so that it's not a very dry, just a bar graph that I'm talking about.
Or numbers in a table.
Exactly, or just numbers in a table. Yeah, absolutely.
The sardine fishery in South Carolina, have they seen that before? I heard something about a decadal change in sea life in sardine fisheries, where it collapses like on a 60-year run or something like that.
That's a great question. So it's unfair to attribute all of this to El Nino events and everything, because it is a really, really complex. I mean, the way that I'm presenting it is just a very kind of surface basis. And I don't know the specifics of exactly how it cycles and everything, but for sure, I mean that the fish aren't necessarily just moving away because it's getting hot or something. And when we see El Nino events, we see rain in certain areas, but we see drought in other areas, or we see very cold events in other areas. So it's not just for our area where we know that the temperature gets higher and that we see these changes, but globally it's very complex.
Thank goodness.
What about overfishing? Do you have the data on that?
That we haven't. What about overfishing? Is the question. Do we have any data on that? And that's something that that incorporating the fisheries experts into is going to be really important moving forward. And we do, overfishing, and especially for a lot of entanglements for animals, a lot of those are fisheries interactions, and we haven't incorporated specific fisheries data, but that's something that certainly could be looked at, because it's something that has to be balanced. And and the U.S. does have quite a few regulations in place, but they're certainly not the only player, right? And these fish don't stay in the U.S. They don't know what the U.S. is, and so some, we know that, for instance, the tuna, that part of their migration is that they migrate to Japan and then back, and they go up to Alaska and everything. So it's something that globally we all need to be aware of for sure.
Is there much data on animals that have been released, their survival or where they've gone?
So sometimes we're able to put satellite tags on animals, especially if we have questions about if they're species that we don't know a lot about, like Guadalupe fur seals. We had a Guadalupe fur seal that we released recently that did have a satellite tag. And we also track the number of animals that restrand, or if an animal dies and they have a tag, then we're able to know what rehab facility they came from, and that kind of information. But the satellite tags are on a very small scale basis. They're several $1,000s for each tag, and and they only stay on until the animal molts and the fur comes off, and so we have a limited data set there. But we're trying to get more and more, because that's a great point. Is that a lot like with domoic acid, if animals are very severely affected, they may act kind of normal, but then sometimes we will get animals that come back in either because they've gotten another dose or because they've had this permanent brain damage.
So at the beginning of the talk, you actually mentioned crowdsourcing, or people who go on whale watching. I know, so the last time we went to a biodiversity festival, National Geo actually have an app where people, I think they call it iNaturalist.
Yes.
Where people can take pictures of different birds or plants and they can input data and it all goes into the system. Is there something similar for whales or marine mammals?
So we actually looked at the question was about iNaturalist, which is an app that National Geographic has. And it's a way to collect crowdsourced data about wildlife in the area. And there kind of have been some small apps that there are some stranding centers in the southeast that have an ability for people to tweet or however that there's a dolphin that's stranded and things like that. And we looked at iNaturalist to see if there would be a way to incorporate some of that data. Right now we haven't really gone into any of the crowdsourced data yet, but the hope is that it's really valuable data that could be used in the future.
Related to dominic acid, you know that it affects humans too. How much of fish, shellfish, people need to eat to actually get sick?
Okay, so yes, the question was about dominic acid. And since it does affect people, how much fish do people have to eat? And it really depends on the amount of toxin that's in the fish. And so the fish will eat the algae. And then the marine mammals or humans will eat the fish. But the domoic acid is a water-soluble toxin, and so it's actually cleared from the body very quickly, within days. And it's the same thing with the marine mammals that come in. If we can flush the toxin out of their system, and they don't have the brain damage yet, then they typically do very well. And so so it's really a question. That's why they monitor levels in mussels, because it could be that you just need to eat one mussel if the toxin is very concentrated, or if it's very low, then you would need to eat quite a bit. I don't have a good baseline answer for that.
There was an article in the Cron calling domoic acid a red tide. It's very good for us.
And there are a variety of toxins. So we know that ciguatoxin affects monk seals, or it has potential to affect monk seals. Brevetoxin, which is the typical red tide, will cause deaths in manatees in Florida. And so we do see kind of hot spots, but they are, people talk about it as kind of an overlapping red tide, but there are a lot of different species that have different toxins.
For Hawaii, is there a possibility with the loss of space for those rookeries where they used to go and the rising sea level to do a preserve on beaches during the pupping season?
So right now, and this is going into a lot more information about the monk seals, but right now the majority of the monk seal population is in the northwest Hawaiian Islands. And so that's a series of kind of atolls and smaller outcroppings and things. And that's actually a national monument. So people are not allowed to go there at all. And then on the main Hawaiian Islands, there are only a couple hundred monk seals out of the 1,200 that use the main Hawaiian Islands. And definitely when they know that certain animals will come back to the same beach, and they'll rope it off if an animal comes to use that beach. And so, but as their habits and their uses change, we're going to need to adapt to that because the main Hawaiian Islands are much more populated. Absolutely. So they try a lot of different things to try to be tolerant of the people that live there, and then but then also to help protect the seals. Thank you guys for coming.
Yeah, thanks everyone for coming out tonight, and thanks so much for your support in so many ways of the center. And thank you again to Claire. This is just phenomenal, and I know everyone's really excited about the health map. So.